Hello, my name is Nain de la Cruz, and in this presentation I'm gonna show you the preliminary results of a research titled Combined Therapy in Breast Cancer, an in silico approach. This work was done in collaboration with Diana Bautista Espinosa, William Albornoz, Jesús Rodríguez González from the Centro de Investigación y de Estudios Avanzados del IPN, and Patricio Zapata Morín from the Facultad de Ciencias Biológicas de la Universidad Autónoma de Nuevo León. The content of our work is divided as follows. First, we present the introduction. After that, we show the methodology. Then we discuss the results. And finally, we provide a conclusion and further work of our research. The base of our research is on the fact that cancer is one of the leading causes of morbidity and mortality around the world. This despite the technological and medical advances we have seen in the past decade. Among the types of cancer, breast cancer is the leading cause of death among women. Therefore, we decided to focus on this type of cancer for our research. One of the techniques used to counteract the effects of breast cancer is using ionizing radiation, which is supposed to induce DNA damage. However, the effects of ionizing radiation upon cancerous cell is limited by radio resistance, allowing so the proliferation of cancerous cells. Therefore, we believe that a better understanding of the molecular interactions which trigger the apoptosis is necessary. But why is this so? Inside a healthy cell, there is a key signaling pathway called p53. Among the activities of this signaling pathway, there are cell cycle arrest, DNA repair, and apoptosis. However, in cancerous cells such as MCF7 cell, apoptosis is never triggered, even if we have the same elements in the signaling pathway. So in this case, the signaling pathway can only trigger cell cycle arrest and DNA repair, allowing the cancerous cell to proliferate. But how this happens? Inside the signaling pathway, there are feedback regulators such as MDM2 and WIP1, which modulate the activity of p53, which is the protein in charge of activating the apoptosis. Therefore, if the p53 protein never reaches some level, apoptosis is never triggered, and the MCF7 cells activate survival pathways instead, such as P21 and AKT. So we see necessary the targeting of regulatory nodes such as MDM2, WIP1, XIAP, BAX, PLAK3, which can sensitize MCF7 cells to radiotherapy. We propose that we can prove different scenarios using a mechanistic approach through a mathematical model, which will allow us to identify radiosensitization strategies. So in our work, we propose to find radiosensitization strategies in MCF7 cells using a deterministic mathematical model. And to do that, we're going to extend a previous p53 model, which now will include key regulators such as PTEN, CCNG1, PLK3, BAX, BCL, and XIAP. After we have generated our model, then we are going to quantify and compare the response of the cell under ionizing radiation alone and under a combination of ionizing radiation plus Nuclein 3A. So, in order to generate our model, it was necessary to run a differential analysis. This analysis allows us to focus on key elements that are necessary for the model. From this, we found that proteins such as BAX, PLK3, CCNG1, PTEN, P21 and WIP1 were necessary in the model. This will allow to improve the biological realism of the model and also to improve the predictive accuracy of the model. Once we identified the elements that were necessary for a model, we generated a block diagram such as the one you are seeing in the figure. And in this diagram, we can see known elements such as MDM2, WIP1, P53, ATM, and also the new elements of the extended model such as PTN, BAX, BCL, PLK3. And also in this model, we can see that we are going to use an inhibitor of MDM2, which is called Nuclein 3A. After we generated this block, it was necessary to translate it into a mathematical model. In this case, we use ordinary differential equations, and the total number of these differential equations were 19, which we used to generate three different scenarios or analyze three different scenarios. The first scenario was the to analyze the response of a healthy cell. In this case, for all the scenarios, we focus only in key proteins such as P53, Cas3, WIP1, and MDM2. For all the plots we are going to show in this section, the horizontal axis indicates the time, which is measured in hours, and in the vertical axis shows the concentration of the protein, which is measured in micromolar. If we see a steady level of the protein, then we can make sure that the apoptosis has been triggered. However, if we don't see a steady level of protein, then the apoptosis is never triggered. So, in order to generate the response of the cell, we use different levels of ionizing radiation, which are 1 gray, 2 gray, 3 gray, 4 gray, and 5 gray. From the results, we can see that low levels of ionizing radiation never trigger the apoptosis, and instead we can see an oscillatory behavior, for example, of P53, which is an indicator that apoptosis has not been triggered. However, if we increase the ionizing radiation level to 3 gray, then P53 reaches an stationary level at around 2 micromolar of concentration, indicating that the apoptosis has been triggered, and this is corroborated by the plot of Cas3, which has reached a concentration level of 0.5 micromolar. If we keep increasing the ionizing radiation to 4 and 5 levels, we see that the time needed to reach the apoptosis has decreased from 45 hours to around 32 hours. So this is the response of a healthy cell under ionizing radiation only. However, when we analyze the MCF7 cells, then we can see that even if we apply a high level of ionizing radiation such as 5 gray, the steady state of the P53 is never reached, and we only see oscillatory behavior, which is an indicator that apoptosis has never been triggered. This is corroborated if we focus on the plot of Cas3, which has a very low level of concentration. But also, if we see the plot of MDM2, we can see that it reaches a stationary level, which also corresponds to the low level expression of P53. So with this plot, we corroborate that our model reflects the behavior of MCF7 cells, which is that apoptosis is never triggered, even at high level doses of ionizing radiation. The third scenario we analyze in our research is a combination of ionizing radiation plus an inhibitor of MDM2. In this case, we kept constant the level of ionizing radiation and we variate the level of concentration of the inhibitor from 0.5 micromolar to 5 micromolar. And we also focus on the same expression of proteins, which is P53, Caspase-3, WIP1, and MDM2. From the simulation, we can see that at low level of concentration of the inhibitor, which is 0.5 micromolar, we cannot see the apoptosis triggering. However, if we increase the level to 3.5 micromolar, then we can see that apoptosis has been triggered. This is corroborated not only by the steady state of the p53 protein, but also by the steady state of caspase-3. However, from the simulations, we see that even if we increase the concentration of the inhibitor to 5 micromolar, the time to reach apoptosis is not enhanced. Therefore, from this simulation, we can see that it is possible to trigger the apoptosis in MCF7 cells, but there is an optimal value of concentration of inhibitor, which is around the 3.5 micromolar. So with this, in this presentation, we focus on showing an updated mathematical model of the p53 signaling pathway to analyze the response of MCF7 cancer cells under ionizing radiation and MDM2 inhibitor, which is nuclein 3A. From the results, we saw that apoptosis can never be triggered in MCF7 cells, only using ionizing radiation. But if we combine radiation plus nuclein 3A, then we can restore the p53 activity, inducing apoptosis. Also, we found that at around 3.5 micromolar of concentration of the inhibitor, it is possible to induce the apoptosis, and it seems from the results that 3.5 micromolar is the optimal value of the inhibitor in order to induce apoptosis, since even if we use a higher level of 5 micromolars of nuclein 3A, the time needed to induce apoptosis was not better. So these are the results, the preliminary results of our research that we wanted to share with you, and we thank you for your patience in our presentation, and we are also open to any questions you might have about the content of our presentation. Thank you very much.